a pasture pest in paradise. All right, so the two-line spittlebug, Prosopia bicincta, was recorded on the island of Hawaii in 2016 on the western coast in the Kailua Kona area, where it had caused a pretty significant damage to pastures. This species of spittlebug is native to the southeast United States, where it has long been recognized as a pest of pastures and grass turf. In Hawaii, the pest has rapidly spread and now infests nearly 150,000 acres and threatens the livestock industry in the state. Kikuyu grass, Pangola grass, among others that make up most of the forage base for the cattle industry, have proven to be highly susceptible to spittlebug feeding and have quickly died out when two-line spittlebug populations reach critical levels. The rapid loss of these grasses creates an environment conducive to the rapid colonization of many noxious weeds, including pomacani, wild blackberry, fireweed, hilo grass, and many others. As a result of the rapid conversion of two-line spittlebug affected rangelands, ranches have been forced to drastically reduce livestock numbers due to the lack of suitable forage, placing a financial strain on the sustainability of their operations. Consequently, this pest poses a major threat to the Hawaii pasture-based livestock industry. Morphologically, the nymphs are smaller than the adults. They lack wings and have yellow, white, or orange bodies, while their heads are brown with red eyes, and they have two orange patches on either side of their abdomen. Two-line spittlebug adults are vividly marked by two orange or red bands that traverse their black wings. Their heads, abdomen, and legs are a deep red color. Adults can jump and fly and live for approximately 4 weeks. Each two-line spittlebug female can lay up to 150 eggs, starting at about 7 days post-emergence as an adult. Eggs are laid at or just below the soil surface near the crown and along roots of the grass plants the adults are feeding on. Eggs can either hatch within the same season or after 19 days, or based on conditions, be laid as diapause eggs. In Hawaii, the pest enters diapause by November with very little activity over winter. Diapause breaks around April or May after hatch. The nymphs immediately begin to feed and progress through 5 instar stages, completing their development in about 50 days, encased within a spittle mass that they create. The complete life cycle of the two-line spittlebug takes about 76 days. Typically, there are 2 generations each year in the summer period. Both nymphs and adults feed on the grasses, albeit at different locations of the plant. The nymphs feed on the roots and crowns of the grass plants. That, to a limited extent, can lead to water stress and decreased productivity of the plant. Nymph feeding alone, however, does not lead to plant death. The adults feed on the xylem fluid and release amylase, which is an enzyme that breaks down starches into simple sugars. As the amylase builds up in the plant, it interferes with photosynthesis and carbohydrate translocation and storage. Ultimately, this results in phytotoxemia, the consequence of which is the browning and eventual death of the plant leaf material. At the very least, two-line spittlebug adult feeding decreases the productivity and nutritional value and palatability of the key forages for grazing animals. In the worst case, though, it results in the complete die-off of key forage grasses and a rapid transition to unpalatable, non-native invasive weeds. So we've been conducting monthly surveys, and the data collected along several of these transects established beginning in 2017 have revealed that the pest is expanding its range at about 35,000 acres per year. Initial estimates of the pest range in 2016, when it was first recorded, covered just under 30,000 acres. By fall of 2019, the pest could be found within a 442,000 acre area, stretching from Puwawa in the north to Honaunau in the south. The monthly monitoring of the two-line spittlebug nymph densities along this same series of transects has revealed key information on the population dynamics habitat selection, and potential ecological and economic impacts of two-line spittlebug on Hawaii rangelands. The four maps in this slide show two-line spittlebug nymph densities changing during peak period of activity from early break of diapause in April of 2019 to July of 2019. The darker red the color, the higher the nymph density. High nymph densities lead to high numbers of adults feeding in an area and often catastrophic die-off of the key grasses. Uh-oh, we just had a power, well, hopefully I don't lose power. Damage from the adult two-line spittlebug feeding can accumulate very quickly under the right conditions and population size. The pictures in this slide show the rapid die-off of kikuyu grass shortly after a two-line spittlebug outbreak and 6 months later in January of 2019. Two-line spittlebug activity was noted in this location starting in April of 2018, and by June of 2018, the pest had caused damage that's noted by the red outline in the picture on the left. The population density reached a critical level later that summer, and by January, all the kikuyu grass beyond the original infestation, covering nearly 200 acres, had browned and began to die. Two-line spittlebug damage goes through stages, sometimes separated by a season. Because of this, the pastures can be a mosaic of healthy grasses, shown here at A, small patches of dead grasses, pointed out by B, and mature infestations that are large and giving way to invading weeds, shown here at C. Early infestations, when adult populations are low, result in small patches of dead grass from adult feeding, shown here at B. These small patches develop in otherwise healthy grass communities when emerging adults from nearby heavily damaged areas, such as at C, move to find suitable feeding and egg habitat. Subsequent generations of adults at B, often spanning 2 seasons, will increase exponentially and reach catastrophic levels that result in widespread damage that's shown here in C. So the next 4 slides show a series of images collected from the same transect, HULT1, and the accompanying graphic of the spittlebug nymph densities collected from May of 2018 to November of 2019 along six different transects spanning the range of elevations. So on this graphic, HULT1 is this dark blue line that you see here. This is the one that we're focused on. This picture is taken at the bottom end of the transect. And that's the viewpoint from all of the pictures in this series. So in May of 2018, the pastures at HULT1 was a typical healthy mid-elevation kikuyu grass pasture comprised of 85% kikuyu grass, Pangola grass, and some Yorkshire fog. 10% white clover and a spattering of various weeds like fireweed. The first spittlebug nymphs were recorded along the transect HULT1 in May of 2018. Over the summer of 2018, the nymph population at transect HULT1 displayed the typical 2-generation growth function that with a peak in June at about 10 nymphs per square meter and a second at about 35 nymphs per square meter in October, before slipping into diapause in November. The plant community changed little over this period. At the November reading, kikuyu, Pangola, and Yorkshire fog still dominated the plant community at 80% of the cover. Clover accounted for 10% of the cover, while weed cover increased by 5% over the level in May. In November, two-line spittlebug activity went into diapause with very little nymph and adult presence over the winter. Over winter, there was a slow but continual change in the plant community composition. By May of 2019, kikuyu and Pangola grass cover had decreased to 75%. White clover cover increased by an additional 5%, and weed cover had held fast at 10% of the plant community. Diapause broke at HULT1 in April, somewhat later than in 2018. And by May, the nymph density was 97 nymphs per square meter and peaked in June at 129 nymphs per square meter before rapidly decreasing between July and October from 76 to 2 nymphs per meter squared. By November of 2019, just 4 months after the peak nymph density of 129 nymphs per square meter, the plant community had completely changed and was no longer recognizable. Grass cover at the November 2019 reading was just 2% and was comprised mainly of fountain grass. Clover cover 2 had decreased from 15% in May to 5% in November. Meanwhile, fireweed cover increased to 93%. So in the space of a year, two-line spittlebug activity reduced a highly productive pasture dominated by high-quality forages, including kikuyu grass, pangola grass, and clover, to a fireweed-dominated landscape with no forage value for livestock grazing. So this Google Earth image of the pasture represented by Transect HULT1 was taken prior to the two-line spittlebug infestation in May of 2018. The red boundary line drawn on the image approximates the extent of the pasture acreage damaged by the two-line spittlebug between May of 2018 and November of 2019. The area is approximately 105 acres in size. Two-line spittlebug damage over this 105 acres resulted in 100% loss in grazable forage and equates to a loss of approximately 420 animal unit months of grazing for the ranch. Now, this is enough to support 35 head of cow-calf pairs for 1 year. This means the ranch will have 35 less calves to market each year, resulting in a loss of revenue of over $14,000 annually. And this is not the only location on this ranch showing this type of damage. The two-line spittlebug research team started a collaboration with Hawaii Department of Agriculture in 2017. The team developed a 5-year work plan with an estimated cost of about $1.3 million. And the work plan objectives include a rancher awareness and education campaign, conducting field surveys for detection and rapid response, development of IPM strategies, biological control agent exploration, and research on the biology and ecology of two-line spittlebug in Hawaii. So far, our funding sources have included the Hawaii Department of Agriculture, who has provided the bulk of our funds, Hawaii County R&D, and the Hawaii Invasive Species Council. We have so far unsuccessfully solicited for federal funding through APHIS and USDA NIFA programs. We recommend an integrated approach to managing two-line spittlebug in rangelands that include limited use of pesticides and other strategies in conjunction with grazing management practices aimed at reducing suitable habitat for the pest. Seeding two-line spittlebug resistant grasses as we discover them will become increasingly important. Recommendations for homeowners who may find two-line spittlebug attacking their lawns are also geared toward first reducing the amount of suitable habitat for the pest. However, given the relatively small scale, non-restricted use pesticides can be effective in controlling nymphs and adults in the lawns. Two-line spittlebug poses a significant economic and ecologic threat to Hawaii. The rapid spread of this pest and its extensive damage to key pasture grasses puts Hawaii's livestock industry at tremendous risk. In addition, the pest poses a significant challenge to the management of and conservation of forest lands, watersheds, and other natural resources. Two-line spittlebug is expanding its range on the Big Island by about 35,000 acres a year. At this rate, it will cover all of the important ranch lands within the next 10 years, if it's not spread sooner by human activity. Our recommendations to the state are to urgently adopt aggressive programs to combat the two-line spittlebug. These include a vigorous inspection and control of the movement of plant and soil materials within Hawaii County and between counties, establishment of a pesticide application assistance program to strategically spray initial small-scale infestations, and a seed purchase assistance program to help ranchers recover and reseed damaged ranch lands. Without these measures, the two-line spittlebug will continue to spread and cause devastating damage unabated, placing the Hawaii livestock industry in great peril. Our two-line spittlebug team is comprised of myself, along with Dr. Mark Wright, Dr. Dan Peck, and Melanie Oshiro, graduate students Shannon Wilson and Robert Sakuda, and research technicians Justin Ye, Elizabeth Whitney, and Yoshiaki Higashidi. Our partners include the Hawaii Department of Agriculture, Hawaii Invasive Species Council, Big Island Invasive Species Council, Hawaii County R&D, and the USDA NRCS. You can direct any questions to me by email or phone. Thank you.
Thanks, Mark. There is few comments, suggestion in the chat box, and actually some reactions in the chat box. So we can take one question for Mark Thorne on his presentation.
Mark, this is Chen. So have you heard any golf course or homeowners that complain about the two-line spittlebug?
So we've actually had some reports of from homeowners about the pest in their lawns and in around their homes. So I haven't had specific contact from golf courses, but I did attend a landscaping conference last summer, and I did talk to several of the landscape groundskeepers for some of the golf courses, and they at that point weren't aware of it, but it's my guess that there's several golf courses right around some of our hottest areas for the two-line spittlebug. I'm certain that they're, the pest is in their garden or in their on their courses, their links, and so I don't know how they're dealing with it. I'm not in contact with that industry, but we have tried to get information out to them. So.
Great, thank you. Yeah, so I'm also keeping my ears open with the turf and landscape industry in case I hear anything. Yeah. Mm.
So Fanny has some message. One message. Bix has also published 2 articles in the Lich magazine on two-line spittlebug. Thank you. So the next presentation is from Cheng. Cheng, whenever you are ready.
Sure. Let me share my screen. You probably need to do that thing for me too.
Maybe not now.
Okay, let me try that.
Yes, can share. Yeah. So just a minute. I would like to congratulate Cheng on behalf of this working group on his achievement on the award on arboriculture research in the year 2019 from the International Society of Arboriculture. Congratulations, Dr. Cheng.
Thank you, Roshan. It was from the Western chapter.
Oh, Western chapter, International Society of Arboriculture. Okay, thank you.
Yeah, thanks. Hi, good afternoon, everybody. My name is Zhiqiang Chen. I'm from Department of Pest. Like Roshan called me Chen, so most people just call me Chen because, as you can see, my first name is quite a challenge to pronounce. So I will be talking about some research updates on some important landscape and turf grass pests in Hawaii that I have been working with and I'm still working on in the last 6-7 years. I have been here for 7 years already. Wow. So we will cover these 4 pests in a little bit more details because, given the limited time, we will only have like 4 or 5 slides for each of the coconut rhinoceros beetle, low-back glass scale, ficus, stem and leaf wasps, and hala scale. And I will have 1 slide for each of the rest of the pests, depending on whether I have time to go through each of them or not. But we will go through these 4 major pests first. So as you all know by now, probably coconut rhinoceros beetle or CRB has been confirmed on Oahu since 2013. So then soon after that, Department of Agriculture, USDA, and HDOA and us, UH, and HISC, we launched a CRB eradication program, response program. So for my research in my lab, we focus on chemical and biological control of rhino beetle. So we did multiple tests, a lot of lab assays, that basically covered, two graduate students' thesis work, and some of the visiting scholar and the postdoc work as well. So we identified multiple low-risk systemic insecticides that are very effective in the lab assays. And then we also completed one large-scale field trial, and we are doing another 2 field trials right now at a golf course on Oahu. And we also identified several effective locally collected entomopathogenic fungi strains. We did quite a few lab assays as well, and did a very small scale preliminary field trial so far. See some results. So here are some of the pictures I want to show you. This is basically a lab assay, typical lab assay, how we arrange the mason jars, and we put the rhino beetle inside. And this is the map, the satellite map of the just completed large-scale palm tree CRB rhino beetle trial. We used 125 palm trees in this trial. And this is the very typical, very severe CRB damage, as you can see. I took the picture in Palau when I visit there to see what they do with their rhino beetle control. And very soon, this picture, I took the picture, then this becomes my favorite CRB damage picture. As you can see how many times this one leaf has been damaged by multiple hits by rhino beetles. I have so many data, but given the nature of this mini talk, so I only show you, just some quick snapshots of some results. This one shows you within 4 hours in lab assays, we begin to see those rhino beetle adults paralyzed by 2 of the chemicals we tested. And when this is basically the result of the long-term, large-scale 125 palm tree field trial. And as you can see, in general, this pink colored line, which is imidacloprid, was resulted the lowest infestation level in general. And we pair that with the trap caught number of rhino beetle in the field. The blue line is a nearby area where we do not have the treatment, the field trial. The trap caught numbers is stable and slightly increasing. But for our area, we treated the area, we can see that the number of rhino beetle caught in that area is decreasing. And plus, we found the dead rhino beetle on the ground in the area we treated those palm trees. By the way, I didn't just grab a dead beetle and put it on the grass and take the picture. It was actually a dead beetle sitting on the picture, sitting on the floor, on the grass, and we took the picture and then collect the dead rhino beetle. So here is latest lab assay results we did back end of last year, and we were looking at additional, new pesticides when they become available, whether that we can, include them for the, large scale treatments. So quick message here is we identified 2 more. Based on so many trials, we narrowed down to 2 more new pesticides that we did not include in the first field trial, but we are including these 2 new field chemicals in the ongoing field trial, 2 field trials in Macarena golf course right now. So we started these new trials back in December 2019. I don't have any, I do have data, but I didn't bring any data to show here. But the key message is we are seeing some effects from the new chemicals we included in these new trials. So talking about biological control, we, as I probably mentioned, we did collect many strains of entomopathogenic nematodes and entomopathogenic fungi from Oahu across the island. The idea is to collect locally available, locally occurring strains so that we can make use of them if they prove to be effective. So then, unfortunately, for the nematodes we tested so far, the mortality rate was relatively low, up to 30%. That was Roshan's previous postdoc work with me. We did get the paper published, but in reality, we did not focus on the entomopathogenic nematodes too much after that, but we are not dropping the idea yet. But we had better luck with entomopathogenic fungi, especially several metarhizium species. We had 4 strains that give us over 80% mortality in lab assays. So that's why here's just a quick result showing you, the mortality can reach 80%, 70% after around 10 days also. Then based on this, we moved forward with a small scale field trial. In the field trial, we had about 40% mortality. Of course, not as good as the lab assay result, 80%, but still, it shows some promising result in the field trial. And we are also growing this metarism on rice, so we can grow them faster in a way that compared to using the liquid formulation to grow the metarism in the lab. That's just some quick key results that about the rhinobeetle work. Another key insect pest I have been working with since I got here in 2013 was low-bed lark scale. So this one was first confirmed on Oahu back in 2012, and we did soon after I joined CTAR, I did a host survey plant, host plant survey, and we found over 111 plant species in Hawaii that can serve as a host for low-bed lark scale. And also our statewide low-bed lark scale survey, we basically it was the first detection of low-bed lark scale on the Big Island and on Hawaii back in 2016 and 2017. Then at that time, the low-bed lark scale was widespread on Oahu, and it's spreading from one of two locations to multiple locations on Maui in the open environment as well. I was just informed by Janice, HDOA staff, that Stacy Chen on the Big Island just confirmed the low-bed lark scale in open environment on the Hilo side. So that was just earlier this month. Actually, I think last week. So this pest usually only infects wood parts of the twigs. They don't really touch the leaves or the flowers or any other parts of the plant, but they prefer to attack the twigs or the small branches of their host plants. In Hawaii, their preferred hosts, based on our observations so many years, they prefer ficus species and hibiscus species. So for our research, one of the early research we did on low-bed lark scale was a large-scale field trial on UH manure campus. We used 20 ficus pentamina weeping banyan and 45 Chinese banyan on UH manure campus for this 2-year study. So we tried 2 chemicals, and we tried trunk injection as a treatment approach. So one injection, in short, the long-term result, research result is that one injection can protect the trees against the low-bed lark scale for about 2 years. So here is the key data that I would like to show you. So basically, for ficus benjamina, I call it curative treatments because in the beginning, they have a very high infestation level to begin with imidacloprid treatment, just one treatment, injection treatment in the beginning. And then we were able to see that the low level, the low-bed lark scale was almost eliminated, but then the low level was maintained for almost 2 years after one injection treatment. So for the ficus macrocarpa, Chinese banyan, I call this a preventive treatment. In the beginning, there was no low-bed lark scale to begin with. And with the untreated or the non-effective insecticide, the infestation level or the population of low-bed lark scale basically took off after, within the 2 years. But with imidacloprid treatment, we can see that we kept the infestation level very low, almost minimum, within the 2 years of the treatment. So right now we are looking at a different, injection approach. So we were using upper jet system. It's very effective, but at the same time, it's also more expensive compared to this more jet injection system. So a lot of landscapers, when I talk to them, they said, "Oh, we don't really can afford injection system, which usually costs like $1,000, or the newest equipment costs $2,000 a piece." So then they were saying, can we use the more jet, the simple one to use to inject the trees? So we actually responded to their request, and we are doing this right now on UH manure campus. By the way, we are also looking at the different chemicals. We are not only doing the new approach, injection approach, we're also testing 2 or 3 new chemicals to see whether they can provide a similar or potentially even better control effectiveness against low-bed lark scale on these ficus trees. So pretty much all the ficus trees, the banyan trees on UH manure campus are being used by our experiment for this research. As I mentioned before, low-bed lark scale, another preferred host in Hawaii is the hibiscus species. So apparently this is a very different plant compared to the huge ficus trees. So then we are looking at a different approach. Essentially, apparently the injection it will not work for this, and we're looking at soil drench and spray with different, insecticides to see whether they can work against low large scale on hibiscus or not. So this was just completed a couple months ago. And in short, the result shows that none of the new chemicals we tested, they were as effective as imidacloprid. We include that as the positive control because we know it worked very well on the ficus low large scale control. So that we will very likely repeat this research to basically make the data, publishable in a peer-reviewed journal. Right now, it's just one round, but we do have a lot of replications. I believe 7 or 6 replications under each treatment, and we had 6 or 7 treatments. That was a brief update on low large scale. And another pair of insect pests that attack Chinese banyan trees, Ficus macrocarpa, are Ficus stem gall wasp and leaf, Ficus stem gall wasp and leaf gall wasps. So this was actually my very first research project after I joined CTAR back in 2013. So what they do is, if you only have the leaf gall wasp, as the damage that caused to the trees are not that bad. So that's why people have, the leaf gall wasp have been here for probably 20, 30 years. But with the relatively new arrival of the stem gall wasp, they give a lot of problems to the Chinese banyan, and they are very specific to Chinese banyan. They don't even touch, say, weeping banyan, which is very much similar to Chinese banyan in many ways. And for example, here, what you see is that the galls, they cause the galls to form, and all these galls on the twigs are causing problems for the tree health. And all the small black dots, those are actually the exit holes. When the wasps become adults, then they come out from the twigs and leave these holes openly to the environment. So we used the 45 Chinese banyan trees on UH manual campus for this research, and we tested 2 systemic insecticide, and we used the trunk injection system to deliver the insecticide. So our key result is that the one injection can protect the trees for around 18 months. If you want to keep maintaining the overall tree health, we recommend the landscapers, the arborists, to re-inject after 1.5 years. Basically, that's what we are recommending right now. So here are some key results. So here, the red colored line is the amamectin benzoate, which was identified to be effective controlling this gall wasp. These are the number of stem galls and also the overall stem gall infestation level. As you can see, this treatment performed better than the other treatment and the control. Then we are looking at, again, similar, basically exactly the same slide I showed you on the low large scale. We are looking at lower cost ways to deliver the insecticide, basically with the Mojet system. And also we are testing 2 or 3 new chemicals to see if they can have a better control result compared to previous method we identified and recommended so far. So the next one I would like to touch on is HALA scale. HALA scale is also not very new, as most of you guys know. It was first discovered on Maui back in 1995, widespread on Maui and Molokai right now. But it's only very limited infestation confirmed at only 3 sites, as far as I know, on the island of Oahu. One of the major, the main infestation site was Chaminade University campus. And another one is about several properties in Waikiki Beach area. And just a couple months ago, Becky, I think Becky is around actually in the meeting, Becky informed me that several HALA trees on UH Manila campus was confirmed positive with HALA scale. Those are the only 3 places we know they are confirmed on Oahu. They are not confirmed on the Big Island, Kauai, and the Lanai so far. So Becky and Derek, before Derek retired, they approached me and they want me to run a small scale, treatment trial on Chaminade University campus and see what we can do with the HALA scale. So we tested imidacloprid with either drench or the injection, and also acid-fed with injection. So basically, imidacloprid was working reasonably well. We had a 70% or higher mortality over 6 months of treatment. So we are looking at to try to get more funding to work on a larger scale replicated research project on this. And in the meantime, we are doing this research on Maui as well with different options of insecticide. So this basically why we're doing this on Maui is on Maui is widespread. For for Oahu, we only have these limited locations, and if I set up this trial on, let's say, in Magong, I don't want to bring HALA scale into that area where it has not been confirmed. So that's why we are running this trial on Maui, where it is already widespread. So there's no concern doing that. However, this gives us a challenge given the COVID situation, right? Actually, we have to do this quarantine forms and everything when we go to Maui right now. My postdoc, he was there earlier this week and he has to go again next week. So then a lot of hassle to do that, but it was already started in the beginning of August, before they put the 14-day quarantine back into effect. So we have to kind of continue the treatment based on the protocol to make sure this is completed. So right now this is ongoing over there on Maui. At the Maui Extension Office, there is a little bit space that we can set up trials like this. That's all that's for the four main pests I would like to go through, and I don't think I have too much time. I just show you what else we do. Oriental flower beetle. So we are looking at biological control and chemical control as well. It's kind of similar approach compared to the rhino beetle work. And we did a foliar nematode control on fern. Foliar nematodes. They are very much a problem for the fern, for the the nursery industry if they grow fern. So we actually tested two. You can call it biopesticide. They are both bacteria-based products. So they were working to some extent, actually, as you can see from the result. And also we did a trial with plumeria rust, which is a very commonly found problem on plumeria, which is a very popular landscape, has a very landscape plant, very important in Hawaiian culture. So we did this and we tested two biopesticides as well, Bavaria and Pseudomonas bacteria based products, and they were working to some extent as well. We also compared this to a chemical fungicide program compared to these two biopesticides. So so far I've been talking about the landscape pest management. I of course have worked on many golf course trials within the past years. So I just give a few examples here. This one is called leaf and sheath spot, or most people just call it mini ring. What's called mini ring is if you're familiar with the turf management, there is a very common disease called fairy ring, which has a big circle, and sometimes you can see the mushroom growing on around the circle. So that is very common. So because of the ring size of this, the symptom is smaller than that, so they give it a common name like a mini ring. So very common problem, a very severe problem for two golf courses on Maui, very high-end golf courses. So we set up a field trial over there, and we ran 7 fungicide programs who replicated research plots, as you can see. And we identified this particular fungicide was working the best among the 7 programs we tested. Another even more common problem for golf courses, even for home lawns and parks, are takeout patch, another very common plant pathogen, plant disease problems. Here you can see the early stage of the takeout patch. You can see some of the patches. At this stage, it is very much treatable still. However, in this picture, at this stage, nothing can be done. There's no way you can bring the grass back. Can you imagine these are actually real putting green? People were playing on this. I took the picture when I was teaching my class, and then the golf course superintendent saying, "Oh, so the golfers they are they're aware of they have limited budget. By the way, this is a city and county golf course. I don't want to name it. It looks really bad back then. So they understand the limited budget they can maintain their course, and this so little they only need to pay to play around. So they don't complain too much, but they still do complain. If you imagine you play golf on this kind of putting green. So they did, at this stage, you have to get rid of the dead grass and the top seed or top dress with sod to basically grow new grass from there. So in this stage, very much treatable. So that's why we set up a field trial on a golf course on the Big Island. So we ran several fungicide programs and the rotation programs. We were able to identify that this particular fungicide resulted in the best suppression, disease suppression for this particular turf pathogen, turf disease. So rubber ant, this is one of my early research back on the Big Island. So what happens is this is not really, this particular ant species is not really turf grass pest per se, because they don't cause damage to the turf grass. However, they are nuisance pest. what happens is if there is a heavy rain today, it's almost guaranteed that they will swarm the next day. And then this particular golf course is a very high end of course. 4 seasons actually, I can just name it. The golfers, they pay a lot of money to play, and for membership, they literally drive their golf cart into the cloud of swarm ants. So really bad. And plus the ants, they are attracted to white colored golf balls, golf cap, and the shirts and golf cart. So it's a really a big problem for this particular golf course and also for other places as well. So we did a lab essay we tried several granular ant baits, and we selected 3 that were working in the lab very well, reasonably well, and we set up the field trial. But unfortunately, the field trial result was not as good as the lab essay result, which is kind of common for a lot of this field research. So then we kind of put a pause on this one. We are not dropping this idea, but we are not doing much on this particular project at this time. Free fly. So common problem for a lot of golf courses for years as well. So in a way that free fly is kind of similar to rubber ant, in a way that at adult stage, it is a nuisance pest. Adult stage does not do damage to the turf grass. However, it's attractive to light colored things like the golf ball, the white t-shirt you wear, the white golf cart you drive, those kind of things. However, it is a two-way pest, I call it. At the larvae stage, it actually causes actual damage to the turf grass. And when the population density is high, they can cause severe damage to golf course putting greens. So we set up this trial at a golf course on Maui, where they had repeated fruit fly problems every year. So then we were able to identify this particular treatment was doing the best among all the 5 or 6 treatments we tested over there last year. This was done in 2019, I believe. So this one is very interesting. So we are looking at suppressing middle grass from seashore paspalum. So this picture is kind of, it's kind of strange. You probably don't know what's going on here. What happens is, I would say, let's say 20, 25 years ago, all the golf courses here in Hawaii, they use middle grass. However, in recent 10, 15 years, there's a trend for some golf courses to switch from middle grass to seashore paspalum for various reasons. However, after they do that, they are facing with a new challenge, is that the middle grass that got replaced or common middle grass, they tend to come back into seashore paspalum turf. If you're familiar with turf grass or golf course, you know that middle grass and the seashore paspalum are pretty much the only two, of course, the turf grass species that golf courses here in Hawaii they use. So, but in this picture, what's showing is there is a very much color difference. If you put them separately, there's no problem. But when you look at this, this middle grass patch inside the seashore paspalum, apparently it is not, aesthetically pleasing. And plus, if you're talking about, the texture of the grass, the golf ball will roll at a different speed, at a different, pattern on 2 different species of the turf grass. So for high-end golf courses, they really like to get rid of, or at least suppress the middle grass from seashore paspalum turf. So you can imagine that, it's really kind of tricky to get rid of one turf grass from another turf grass. So I received a 2-year funding, and I had Dr. Judy Frank as my co-PI on this project. We hired a master's student who worked on this project as his master's program. We did, the lab, not lab, open bench test to make sure, to try the different herbicide programs to make sure they essentially they give more suppression. They cause more problem to the middle grass, but they cause very little, a minimum problem to seashore paspalum. And then based on these trials, we set up a couple of field trials at golf courses on Oahu, and we were able to come up with several recommendations that people can use to suppress the middle grass from seashore paspalum. So I'm almost done. Basically, I would like to acknowledge all my previous and current lab members. Without their contribution, we cannot do all of this work. And another quick update is that after Dr. Mike Kawadi retired a couple of years ago, I took over his R4 program as a PI for that program. And in this capacity, I work with Julie and James on specialty crop pesticide program. So we are looking at testing different new pesticides against some important pest problems on not really turf and landscape, plants anymore, but in this capacity, we work on papaya, on coffee, basil, and pineapple, and other things. Acknowledgement for funding agency for collaborations who contributed to all this research. Any questions? I know I'm running this so fast, but maybe one or two questions. Roshan.
Thank you, Cheng. That's lots of information. So we don't have time for the questions also. If anybody has questions, please feel free to type in the chat box for Dr. Cheng. And we will need to move on to another presentation from Brooke Manke, which is eradication effort of little fire ant on Maui. Brooke, whenever you are ready.
Okay.
Okay.
Right. Can you all see that?
Yes.
Okay, great. Yeah, sorry about my presentation, or my video doesn't quite work. I don't know what's going on. I think it's a driver issue, but here we go. My name is Brooke, and I work for the for Maui Invasive Species Committee, and I function as a little fire ant coordinator. And we've worked closely with Hawaii Ant Lab for several years, and our goal on Maui is to eradicate little fire ants. So the presentation today, I'm going to talk about what's up with fire ants, what's going on, how's things on Maui, how we do things on Maui, and then I'm going to talk story about the helicopters, because I think that's how you contacted me, Roshan, as you saw that Vice video that they did on the work we're doing with the helicopters. So I made some assumptions about this crowd, and I think you know probably a lot about pests already, so I'm going to briefly go through this. There's about 50 ant species, or 60, I think. I don't know if this is right. Allison Wagner, if you're there, you can correct me.
More than 60, but you're doing great.
Thanks. So more than 60 ant species in Hawaii, and we have a couple of fire ants. We've got the little fire ant, which is Westmania aeropunctata, and we've got the tropical fire ant. And that's important because tropical fire ants are very different. They don't have the same impact that little fire ants do. We get a lot of calls for tropical fire ants stinging people, and we have to figure out if it is, go send somebody to do a sample, etc. It causes a lot of extra work for us. We also don't have red imported fire ant, and we don't want it, but our surveys would probably discover them. So it's really important that we do follow up with any reports of stinging ants. So that's what a magnified view of little fire ant looks like. The name fire ant is not the best because there's so many fire ants. I wish we had gone with electric ant. That's what they call them in Australia. They have a bunch of different names. They're one of the world's worst invaders. They're arboreal, meaning they live in trees, and they form super colonies, which means it's just one gigantic colony of little fire ants stretching out. That they do give painful stings, and they're devastating to the environment, agriculture, and quality of life. So they sting, that's lame. They ranch pest insects, that's also lame. They sting pets' eyes, lame. And so it makes you wonder if LFA only stands for little fire ants. It may be an acronym that means something else. So they're originally from South and Central America. You can see here, this is from a paper, the green area being their original range, and they have spread out all over the world. They're a real pest in a lot of different places. This is needs to be updated. Maybe Allison, you could send me some updated map here, but this is 2011 on the Big Island. So they're already very widespread and out of control by 2011. I think they were first discovered in 1999. Is that correct, Allison?
Yep. First reported or detected.
And yep, 1999. And so by the time they were discovered, it was probably already very widespread on the Big Island. So on Maui, our first discovery of little fire ants was in 2009. And this is a timeline showing the number of incursions over the years. So the Department of Ag works at the ports, and they intercept little fire ants coming from the Big Island frequently. But this is showing when they've escaped beyond the Department of Ag, and that's when Maui Invasive Species Committee comes into play to work on eradicating naturalized populations that slip through our security, our biosecurity. And so right now I have 15 incursions discovered to date. Might be 16 actually, but here, yeah, it's 16. I need to update that slide. There's a map showing all of the locations on Maui. And in red, I'm showing active sites. Those are sites that we are currently treating. They're in the first year of treatment where we treat every 6 weeks, and I'll go into that in a minute. The yellow sites are transitioning or spot treatment. That means that they've had a whole year of treatment, but they continue to have little fire ants in pockets or spots. And so we adapt our strategy at that point to go just treat those specific locations instead of the entire site. And then the blue are sites that are in a monitoring phase. That means that the last survey or last several surveys, we did not discover little fire ants. And so we are monitoring, and I'll explain that process in a moment. And then here are 4 green lightning bolts, and those are what I'm calling an isolated incursion. So the ants never appeared to naturalize in those locations. One of them was like a big box store that brought in hapu'u, and they were treated and they were never discovered again. Same as a nursery. One was a pallet of shingles that came from the Big Island that was infested. It was identified and quickly treated, and they've never showed up again. And another was like a vehicle that was moved from an existing site, discovered little fire ants on it, treated, and we go back and continue to monitor that location, and there never has been little fire ants since the initial treatment. So those infestations widely vary in size, and as you can see, there's one very large infestation. That's the Nihiku infestation that we're using helicopters, which I will talk about at the end of this. I've also color-coded these. I need to update their colors, but they were at one time color-coded to the map that I just showed about monitoring and active treatment. Several more of these are in a monitoring phase now. So little fire ants, it's important to know that they spread slowly by themselves. A lot of ant species will create a queen, which will fly away, maybe a long distance, and start a new colony. But little fire ants don't do that, not here in Hawaii at least. They spread by budding, where a queen will just crawl away, maybe just a meter away, and start a new nest. And so the super colony is comprised of nests, many, many nests all over the place. And it's also important to know that it must be a queen to establish a new population. Just because you have several workers does not mean that you are now infested with little fire ants. And the biggest risks are movement by people and by waterways, which has happened here a number of occasions, but probably accounts for the large size of the Nihiku infestation, which again, I'll talk about later. And when water moves little fire ants, the queen has to survive that event and likely has to have workers to help reestablish that new colony. So what we're finding is it doesn't happen as frequently as one might expect with the waterways. So here I've created this graphic. I give a presentation similar to this to the public a lot when there's a new infestation in somebody's neighborhood. And so this is to give a good idea of what are risky behaviors or not. So like if you're just walking through an infestation, you're not going to spread little fire ants. Or picnicking, maybe you could possibly, it's a little higher risk. Or using garden equipment. So there might be clods of dirt or something if you don't properly clean it. But the biggest threats are moving debris and green waste and dirt and or idle materials. So like I described before, a derelict vehicle that was parked on a property for a long time that had little fire ants established on it, then that vehicle was moved. So those are all important things to know, especially for the public. So what do we do for little fire ants? We got to go out, and first thing you have to map and delimit that population, meaning you have to find the boundary of where that population is. And that's a difficult process. So we take vials or sticks with peanut butter and put them out for about an hour. We come back, pick them up. If it's got ants in there, we put a cap on it. We put a number on that cap. we take a GPS point with that number, we put them in a bag, we take them to the lab, we look under a microscope, and then we can properly identify the ones that are little fire ants. There are several look-alike ant species, and they are so small you cannot make a positive ID with your naked eye. Some may counter that claim, but that's how we do things. We make sure that it's positively ID'd under the microscope. And then the way we space our samples, of course, matters. So the tighter the spacing you can do, the better, but that's a lot more work. So we generally go with a 5-meter spacing, which correlates with the what we believe to be the range, the foraging range of little fire ants. And sometimes we space it out if it's a very large site and we can't do that sort of tight spacing. And this is what it looks like. So if I were to drop a sample there, I would be surveying an area of up to 5 meters in all directions. And so if there's any nests in that area, I would detect little fire ants with that sample. Those nests in that area have a range of 5 meters. So that would, this here you see is the area that they are probably operating in. And so when I calculate an infested area, I am using 10 meters from every positive ID, and that's sure to encompass the ants that we might have detected with that positive vial. When we do treatments, we do a treatment out to 20 meters. That's to ensure that we're not missing anything. Our sampling process, of course, is not perfect. And so if there was an ant that was beyond where we sampled, we would be putting bait out into their foraging range and hopefully getting that ant. So once we have calculated a treatment zone, we often will make a grid. This is a 10-meter grid, so you'd want to put a sample in the center of each one of these squares or somewhere in each one of these squares. It's not always easy or feasible. Or we can do a 5-meter grid. This is what a 5-meter grid would be. So there's one positive ID. This is all the areas that you'd want to sample in that 20-meter treatment zone. And that's what we go back and sample over and over again at all of our infested sites. So when we do treatments, we do multiple types of treatments. You can see here, this moving image is the gel bait that Hawaii Ant Lab developed. And you can use a toxic bait in there, or you can use an insect growth regulator. The insect growth regulator is birth control. If you continue to feed this to the queens, then they will stop reproducing and eventually the colony will collapse. Workers die of old age. The queens don't have those workers tending to them and feeding them any longer, and they have to go out and forage on their own, put themselves in danger, maybe die. So the other types of baits we use are toxic, and we can put that in this gel bait. The gel bait is really important because it can be squirted up high into the trees where the ants, like I said before, they live in the trees. They can have colonies up there that never have to come to the ground. They can get all of the resources in those trees. And so to get rid of them, you need to be able to get your bait up into the trees. One thing about bait is that those workers, they go out, they eat that bait, and they bring it back to the queens. So the toxic baits, there are, we can put in a gel, but we can also use granules. And so down on the bottom left, you see there's somebody squirting gel bait up into the trees, and then someone using a blower to blow granules out. Those granules we use generally when it's open area, like turf or without overhead vegetation. And I've just put some relative toxicities in here. You could probably find a lot of problems or issues with this graph, but the baits and the toxins that we're using are relatively low toxicity when compared to a lot of regular common household items. When we do the work, we use a tablet with a live GPS. We're able to take the data that we've collected, put it right on there, and that helps guide us with the grids, like I said, so that we can see in real time if we are covering the area that we really need to be covering. It's very important to be totally thorough in invasive species management, as I'm sure all of you know. We use a chest harness to have that hands-free data right in front of us, showing our progress real time. The Control strategy is when we delimit an infestation, we have a year of treatments ahead of us every 6 weeks, and we start the first 6 months with that insect growth regulator. We feed that birth control to the ants for 6 months, at which time we switch to toxic baits. The idea being that after 6 months, you can be fairly certain that most or all of the queens are on birth control and therefore not reproducing. So when you switch to those toxic baits, it's like a 1-2 punch, and it really is more effective than just using either or. So at the end of 1 year, we go back and do a very thorough survey. And if there are no ants, we will switch into a monitoring phase. And we have set a threshold of 5 years. And for 5 years, we will do 10 surveys. In the first year, we do 4 surveys, the second 3, 2, 1, and the final year we'll do 2 more surveys. And if there have been 0 ants during that entire time, we will use the E word, eradicated. And only then do we say the E word. Of course, at any time during that, if we find little fire ants, we go into a spot treatment phase where we will treat just that specific area, and the monitoring process will start over again. So what does that look like? This is a real site on Maui, and this was the delimitation effort that occurred there. So I'm going to walk through all of this data, how this works. So we're most interested in the positive IDs. All the blue dots that just disappeared were not little fire ants. And so this would be the infested area, a 10-meter buffer of those positive IDs. This would be the treatment zone, a 20-meter treatment zone. So we put that onto our GPS tablet so that we can have that as we go out and treat, and we'd run a track and we can fill in that area. And as you can see here, this was before we had this tablet system in place, and there was quite a bit missed. But as we moved ahead, we started using the tablets, and now we're doing more thorough treatment. There are sections you can see that are missed, but those are largely big mansions, sections of road, or the beach. And although the little fire ants can live on the beach, we can't treat below the water line, and they generally are in the bushes against the sand. So this is several treatments. There's 4 treatments. This time we usually do a mid-treatment survey to see how things are going. And you can see here that at this mid-treatment, there was only 1 positive hit. But we need to do our full complement of treatments for the whole year. And so we continue on with the treatments. At the end of the year, we did a very tight survey. This time I'm using hexagonal grids, which fit a little tighter. And you can see we did our best to survey all of that area. It's quite an effort to go out there and survey this acreage. And you can see there's still ants remaining in 2 locations here. So this is when we would shift to a spot treatment. Now we have a new treatment zone, and we would go in and continue to treat those zones and survey. This is an example of what you can do as a hybrid grid. You can still do a loose survey everywhere and a tight in the areas that you're focusing on. We change up as needed. And you can see here the areas that we treated, now they're gone, but the ants have reappeared in another location. And so some of these sites can go on and on like this. But not all of them, and several of them are quite a bit easier to control. So that's an example. Here's an example of our original infestation that came back years after it was treated. And this is what it looked like in 2016. And this is what it looked like in 2019. And since then, we're almost to the 5-year mark of finding no ants there. We're continue to do surveys like this, of this nature, very tight around the area. And we believe that this site is probably eradicated. But we won't say that until we reach our 5-year threshold and have completed all those surveys. So here's, again, Maui, the map I showed earlier, but I promised I would talk about the very large Nahiku infestation. And so this is what that infestation looks like. Little animation. It's very large. As you can see, it goes down a waterway all the way to the ocean. That's the treatment zone that we set aside to work on for that infested area. This is animated in Google Earth. Here is some of the data that went into that. You can see there are gaps in this data. So if I buffer these things, this is a 20-meter treatment zone. And then the blue is just a 5-meter buffer of vials, samples that we set out that did not detect little fire ants. This is Malka, and this is probably where the little fire ants were introduced and went downstream all the way to the ocean. That's why it's such a linear and large infestation. You can also see there's gaps, and that is very dense howe and other vegetation that makes it nearly impossible to move through and impossible to do the typical work that we do on the ground to eradicate. So it became evident that we needed something else, and that was a helicopter, ultimately decided. So because of the extreme terrain, dense vegetation, etc., we needed a helicopter. But we also knew that our product, whatever we put out there, was going to land in the waterways. So we looked at several different scenarios of what we should be treating with the helicopter. And because we can't be certain of the edges of the infestation, we needed to make that treatment zone larger than just the 20-meter zone. And so we discussed those, and we were also needed to figure out how we were going to do this with a helicopter. There are existing platforms. There are granules that you can, hoppers that contain granules that you can drop, but the granules that we use are toxic, and they also don't stick in the treetops, can be very expensive, and those toxic granules would get into those waterways, and we cannot put toxic bait into the waterways. Another option that was available already out of the box is a boom spray. You see in the left image, that would be for like crop dusting, etc. And so we considered using that with our gel bait and the insect growth regulator. The insect growth regulator is what we had to use because it was going to go into the waterways, and we had to get a special use permit to do that. So I'll cover that in a minute, but oh, here's my slide. So it's a low environmental impact. It's widely used in the US to control mosquitoes. It's directly put into waterways, especially places like Florida. It's recommended by the World Health Organization for drinking water in regions with malaria. So if you store your drinking water in a clay pot in Africa, they recommend you put methprine, S-methprine in there to control mosquitoes. So it's not really damaging to people, and it's a low impact on the environment. And so we can use that in our gel bait to use from the helicopter, which we need to stay in the trees and go to the ground. Here's this slide again. As you can see, I was a little hasty. So this is what the gel bait recipe mostly comprises of. It's mostly water and vegetable oil. It's got beef liver powder. That's the protein that attracts the ants. And then the product we're using is called Altocid, and it's 2% is the active ingredient, the inactive ingredient, and then the methylprene is 0.5%, so a very low percentage. The other ingredient we use is xanthan gum, which is a thickening agent to make it into the gel that allows it to stick in the trees. This was developed by Hawaii Ant Lab. And so to get ready for that, this was discovered in 2014, and we only began treatment last year. So there was 5 years where the ants were this whole infestation was not being treated. But what we did do is we treated the high traffic areas. So the roads around people's houses where we thought, again, people would move the ants, because that's what's going to move the ants the quickest and the furthest. The budding, the way they naturally move by themselves was less of a concern. So we had to get a special local needs permit to use the Altocid in this area. So there was an inventory for threatened and endangered species, and U.S. Fish and Wildlife came out and did a report. One of the concerns was a native damselfly, and it was not present in the area. And then we had to figure out what we were going to use. We're going to use a boom spray to do this. And we also had to figure out how we were going to get this here because you can't readily buy it in Hawaii. So we decided we'd try out that boom spray. And we had a lot of tribulations with this. Here you can see the gallons going down because of the battery that we were using to operate the boom spray was losing juice over time. So we had to trash this test. And we went back and we fired up the helicopter so that the battery wasn't an issue, and we ran our gel bait through these booms. But with that big system and that gel bait, it does not act like water. So we were pushing this gel through something that normally uses a very liquid material, and each one of these nozzles had a different flow rate, wasn't pushing out what we needed. But on top of all of this, the helicopter had to go a minimum speed of 45 miles per hour to avoid the updraft and get the bait all over the helicopter. Because the booms are so close to the helicopter, the rotor wash brings bait back into the helicopter. And so had we used this and flown less than 45 miles per hour, which we need to do to be very specific to the treatment, we would have been coated with this disgusting gel bait. So we had to trash that idea as well. Well, at Maui Invasive Species Committee, we're very familiar with using a long line and a spray ball. And so we decided we would try out our existing system. And so we tried it out. This is the spray ball unaltered, the one we use for plant control. And it showed some promise, but it was not putting out the amount of bait that we needed to be able to treat the entire infestation in a reasonable amount of time. So we needed a new rig with a bigger pump, and we set about developing that. And we bought a new, bigger, better pump, and we tried that out, and there's our gel bait there, and it really cranked out that gel bait. Let's see here. Is he gonna squirt it? Here it goes. Yeah, this is the initial phase of trying to figure it out. And there it goes. It's really blasting bait out. So we also needed to put that bait out in several directions. So you can see here, we've got 5 nozzles. And now we've got the power to push the bait out at a reasonable rate and distance. And so this is what our finished product looked like. That's a modified spray ball with 5 nozzles pushing bait out from the helicopter. One of the issues we had was we need to know how fast the bait's coming out to be able to calibrate it so that we know how fast to fly to get the desired amount of product down over 150 acres. That's the area that we're treating. So one of the issues we had was when we were on the ground running this, it wasn't putting out nearly as much as when it was in the air because of the gravity assist. So to calibrate it, we had these poor guys go down there and hold a garbage can underneath the spray ball as the pilot lowered it down in there, let it go for 10 seconds, and then they ran over and weighed it, dumped it out, brought it back, and we did this over and over again to figure out what the flow rate was from vertical, because it had to be vertical. That line had to be vertical to get the actual flow rate that we were going to be using. We considered going to some bridges and hanging off the bridge and trying to do that, but since we had operations going out of our base yard here, we had the helicopter contracted already, we decided to use the helicopter as is. And so this is how we calibrated it. Then once we had a calibration, we needed to figure out what the spray pattern was. So we hovered, sprayed. We had guys on the ground who immediately ran up afterwards, and they marked the spray pattern with flags. And so we were able to calculate the spray diameter. And so knowing those things, the diameter and the rate that it was coming out, we can then calculate what our speed has to be. So here's how the math was done. What we figured out is we needed to fly at about 10.5 miles per hour ground speed to be able to deliver the desired amount of bait. And so that was acceptable, and we began treatment. So I showed you this earlier. These are some of the ideas that we had. We eventually settled on a treatment zone that looks somewhat like this, and we went to work. And in a helicopter, this is what it looks like. We've got somebody sitting there next to the pilot, and they're starting and stopping the spray with the pilot. As he starts and stops that spray, they're collecting a point, and we're running a track there. That way we can see our progress. You can see that this is colored by velocity or speed. So the closer to blue, the slower. The yellow is a faster speed. And as we cross the edge of the area that we're trying to treat, we take a point, and this is using Locus Pro on a tablet. And that's how we've done it. We've done 8 treatments so far. We have 1 more scheduled, and it will be 1 year of treatments. And the data from a treatment, what it might look like, is this. After you go, and those are all the greens are where we started spraying. All the red dots is where we stopped spraying. And when you process the data, here the yellow lines are showing where we treated. And I'm pretty happy with these results here. And you can, when you collect it with the GPS, you could get altitude data. And so you can see in 3 dimensions what that work looked like. I just think that's a cool visual, so I'm showing it here. And so I do have a video here that shows the whole thing, but it's looks like I'm running out of time and I won't show it, but you can always go to the Vice video and see what it looks like. And probably the question on everybody's mind is it working? And the answer is maybe. I don't know yet. What we need to do is go do an exhaustive survey of the area, which we have not done yet. We want to complete this last survey. And we have done some surveys in the area, and what it looks like is definitely in the open areas where the bait can make it to the ground, it is working. But in the areas where there's canopy, we're worried that not enough bait is getting through the canopy to the ground. And so it may not be totally effective. But again, for that information, you're going to have to wait until we've completed our survey and can give a full report back. So anyways, looks like it's 2:30. Thank you very much for listening. And the Hawaii Ant Lab and Department of Ag, Hawaii Invasive Species Council, and the County of Maui and our crew here are all integral parts. This could not have happened without them. Thank you very much.
Thank you, Brooke. Very interesting presentation. And but we don't have time for questions. If you have questions for Brooke, you can type in the chat box, and we will be staying for maybe another 20 minutes here in the meeting. And so our final agenda is
Yeah, if anybody wants to ask questions, I can stick around too.
Mm-hmm. Or email to any of our presenters today. Brooke, are you sharing your screen still?
No, I stopped
Okay.
the share.
Okay, okay, thank you.
Yeah.
So our last agenda is any new detection or pest alerts if we have in our counties, island, or in any localities.
Hey, Lashawn, this is Springer K. Can you hear me?
Yes.
Okay. So Dr. Chang already mentioned it, but wanted to give a few more details on the low bait lac scale that was detected here in Hilo. So that was just the last week of July. It was on Melotus philippensis, which is a euphorbia, a spurge. It was a small tree, and it was treated within a few days with a soil drench of imidacloprid, which seems to have worked beautifully. The scale just fell right off. We found a single scale on the MacNut tree immediately next to it, but H2A is continuing to do surveys in the area and will let us know if they find anything else. And I think that's it. Just want to give those additional details. Thanks.
Okay, thank you. It's a very good information. So I have something to share with, it's just a little bit of, it might be just a little bit of entomology quiz. If you can see, there is a larvae on the kale leaf. This photo is from one of our cooperative farmer, Martha Whitlock from Molowa Organica. So it doesn't look like an imported cabbage worm and also not cabbage looper. So what do you guys guess? You can type in chat box or shout out. So this is actually a parasitized larvae of some Noctuidae larvae. You can see some cocoons by the side, which is the cocoon of parasitoid. And so as I mentioned, this doesn't look like imported cabbage worm or cabbage looper, not diamondback moth larvae. So I guessed it might be something else, something new. And I sent this photo to our Dick Suda, and I and him guessed it might be something cabbage moth. It's called Mamestra. I forgot the scientific name, Mamestra brassicae or something. But to do some confirmation where we need to get more larvae and rear out the adults. So, but our farmer got only one larvae in his farm of this this kind of larvae. So yeah, so we are on lookout. So this might be just a kind of a alert. We might get this larvae in our farms here in Kauai or somewhere else on other island. And just to know, this cabbage moth is the pest of brassicae only in the serious pest of brassicae only in the Europe, not in continental United States or in Pacific Island. So just to make a little bit of alert, but we haven't got another larvae yet. And that's it from me. So James, do you have something to share with us?
They're more disease updates than pests, but still invasives. Unfortunately, we have a meeting of the ornamental agents with a big industry group going on the same time. So I'm going to kind of combine a few of our different projects and just give a quick update. We've discovered a few new diseases of different ornamental plants in the last few months. Over here on Kauai, we just discovered a type of anthracnose affecting Costus barbatus. It doesn't seem to be affecting all of the Costus, mostly just that species, but we did see some spots on surrounding Costus that other types of cut flower ones. We're working on kind of trying to get some ID work going on it, but it seems to completely ruin the flowers for cut flowers. So we're trying to get the word out there so that folks can keep an eye out on it. Russell Galanti has been doing some work with ring spot on tea plants. It's a virus. He's identified a bunch of different viruses. This one seems to be the only one that's actually causing a disease. And they're working right now on doing some cleanup work, kind of like Mike Meltzer was doing. But they're identified it so far on Oahu, Maui, and Big Island. And they're looking to see if people find it on other islands, so kind of keep an eye out for it. And it seems to be spread by a really wide range of insects, aphids, mealybugs, mites, whiteflies, and then also through vegetative propagation, so manual spread. Hannah Luchin, the ornamental agent over on Maui, is starting a project now looking into protea diseases. There's a bunch of kind of unidentified diseases that seem to be affecting things from the soil level. If anyone's interested in that or has some institutional knowledge, she'd really appreciate some reach out with that. And then I think Russell's talked with this group before about it, but he's continuing his work on a virus and fungus complex causing rapid death of red ginger for the ornamental industry. And so he's kind of been expanding and fine-tuning some of his work to figure out what's really going on there. But he's hopeful to have an update for that sometime soon. So unfortunately, lots of new diseases for us to kind of deal with, but I wanted to get those out there so that if any of you do encounter them, either in your work or even in your yard, get in touch with one of us or get in touch with me and I can pass the info along. Thank you.
Thanks, James. Franny, do you have something to share with us in this meeting?
Nope, I don't have anything. Rashawn Springer handled it.
Welcome. Anybody else who has something to share on new detections or pest alerts?
Hey, Rashawn, I was just wondering if anybody had any updates on the Queensland longhorn beetle on Hawaii Island.
I guess I could.
Okay.
That,
yes, Fanny.
Yeah, I mean, there's a couple of new things going on. We had a few more detections in Ponta Eva farm lots. So we had detections in the Hilo area before, but it had mostly been in down in middle Puna, I guess. But it's moved up a little bit. There's been a number of those, and USGS is going to be launching a project doing surveying out in the area. They have a survey team now in place, but they're still working on their protocols. And
USGS, not ARS.
No, the ARS project is still ongoing, but this will be Bob Peck and Helen so far have launched a project looking at the expansion of a new invasive species. So they have couple, you might know Jordan from, old ROD team member is now joining that team. So they have a little team they're putting together, and I think that's going to be launching in the next couple months. So we should have some more idea. They're looking at population dynamics, and ARS is looking more at the genetics. And so far, the genetics that they've done point to one single introduction of that original beetle.
Thanks. And I'm sorry, for those of you who are not on this island, haven't been paying attention, Queensland longhorn beetle is a new, relatively new, very large cerambycid that when it has attacked has been very destructive to orchards and crops. It has a very wide host range from citrus to coffee to cacao to tea to even palm trees. So it's only a very limited range. It doesn't seem to move very fast. And again, it was one of these things that was lurking here for years. Probably 7 or 8 years before it started causing a bunch of problems, and it's apparently going to be a pretty difficult thing to control. Fortunately, it doesn't seem to move very fast, but where it's hit, it's been pretty severe. I do not know if it has been found anywhere outside of Queensland. Michael, it's a big cerambycid. You can go on BISC's website and see pictures of it. Thanks.
Okay.
Thanks, Franny.
Thanks, Franny. Thanks, JV. Any other new updates? Do we have Jane Anderson here still? Jane? No, not. So, Tiffany, do we know how many parakeets are killed by now?
That's a great question.
Okay.
Jane can give better update on that number, but it has been one of the silver linings with the whole COVID-19 shutdown on Kauai and tourist industry is, I believe it's over, I don't want to give the wrong number. It's over 3,000. It may be all the way up to 5,000.
So last time when we met, Jane mentioned it's 3,500, I think. So yeah, just curious. That's very good then, if it is 5,000. Because we have estimated population is around 10,000, yeah? So half of them
That's a
already
really
gone.
low estimate of the population, but yeah, that was
Oh,
the last
okay,
estimate.
okay.
Yeah.
So yeah, we can just estimate, yeah. So it's 50% already gone, which is very good. So yes, thank you so much.
You muted yourself, Rashawn.
You're still muted.
I'm still muted. Okay, thank you. Thank you so much, participants and the speakers. And just wanted to make a last announcement that if I would wish to run this meeting as a mini conference every 2 months, and it will be in this format, maybe 2 talks and half an hour of discussion. Or if we have 3 talks, that's fine. We will need to stay for a longer time. So 2 talks and half an hour of discussion and 1.5 hours mini conference every 2 months. And I would like to ask if anybody is interested in giving a presentation or talk next time, which will be October, sometime October, mid-October. And you are very welcome. So yeah, if not, I will be contacting you or anybody from outside personally. And thank you again. Aloha and mahalo, and happy, so it's a weekend, so happy good weekend. Have a good weekend to everyone. Thank you.
Thank you. You too.
Bye-bye.
Bye-bye.
You.
Good job, Roshan, and
Thank
everybody.
you.
Thank you very much.
I'm muted now.
Bye, Yushan. Thank you.
Bye. Hey, I can see Adam, Rosemary. Adam, can you hear me? So otherwise, yeah, I will just communicate in the email. Thank you.